Today we're going to talk about DNA replication. Replication means to make a copy, and in this case we're going to be making a copy of a strand of our DNA. Some of the reasons why we might be doing this is we may be carrying out mitosis in order to make new cells to grow as an organism, or in order to replace dead cells, or we may be carrying out meiosis to make sperm and egg. Before that cell division begins, we need to make copies of our DNA. So we would carry out a process called DNA replication. During this, one of the strands of DNA is used as what we call a template to build what we call the complementary strand. Now a template, some of you may be familiar with templates with art, but you may have a template that helps you to draw a perfect circle. And it would help you to build the same circle every time that when you draw it. It's used as a guide. And in this case, we know our strands of DNA are complementary to each other, meaning that if there's an A on one side, there's always a T on the other. And if there's a T on one side, there's an A on the opposite. In this case, we're using that one strand when we separate it in order to be able to figure out how to build the opposite strand. And we do that by using our base pairing rules. This process is going to occur in both directions along a strand of DNA once we've separated it. And I'll show you that in a second. In order to do this, we separate the strand, and it's going to produce two new complementary strands from an original one. So it may sound confusing at this point, but let's take a look and see what it actually looks like. When we go to copy our DNA, if you can remember, our DNA is a twisted helix. It's a spiraled ladder. And in the center are our bases that are paired together and connected by hydrogen bonds. In order to copy the DNA, the first thing we need to do is we need to use some enzymes in order to do what we call unzipping. It's almost like unzipping a zipper. We unzip the strands, and when we do that, we're just breaking those hydrogen bonds in the center and separating them so that we can have the two strands apart from each other and not connected together. The enzyme that helps us with this is helicase. It almost kind of even helps you remember what it does. We know ASE is an enzyme, and helic or helix, right? It's that twisted ladder. When we do that, where we've separated is called a replication fork. And each strand now is going to serve as a template to build the new strands. Remember we said before that the template is a guide. So one strand may have an A. We know that when we're making the second strand off of it, it should have a T. The opposite side had a T, which now we're going to pair an A. So we're going to be building complementary strands to each other. In the end, we'll have two identical strands. The enzyme that helps us to do this is something called DNA polymerase. And notice I put a box around the word polymer. And the reason why is we've talked about polymers before. We know that it's a long chain of repeating units. And in this case, our repeating units are the nucleotides that we're putting together to make the DNA. So if we break this word apart, the ASE tells us an enzyme. The polymer tells us that we're going to be building a long chain of repeating units to make our DNA. So the DNA polymerase is going to join the nucleotides together in order to build the DNA, and it also is going to help proofread. Once it starts to build it, there's going to be another DNA polymerase that comes along in order to look to see if it made any what we might call spelling errors. It's going to look to see if there's an A on one side, there should be a T on the opposite side. If not, it's going to work with other enzymes in order to break it apart and fix that spelling error. At the end of DNA replication, we will have two new strands of DNA, you can see here, identical to each other and identical to the parent strand or the original strand. And that was all because we used the individuals as templates and we used those base pairing rules.